Today I have some really cool news about the evolution of the amniote brain. A new study published in the journal Science found some amazing similarities that reptile brains have with mammal brains, as well as some important differences. The researchers who conducted the study work at the Max Planck Institute for Brain Research in Frankfurt, Germany, and their findings have been described as unexpected and a milestone. The topic of their research was the cerebral cortex in amniotes, which is a large portion of the brain that's responsible for a huge amount of data processing. The cerebral cortex is involved in, among other things, awareness and perception, in abstract thought, and in processing language, and it also stores memories. It does a lot, but we still don't have the full picture. There's a lot of debate over how the amniote brain evolved in the first place, like how and when a particular brain structure grew or shrank in size in a particular animal lineage. To settle these debates and to answer their questions, researchers have to study the brains of all kinds of amniotes, be they reptiles, birds, or mammals. And after gathering all of this physiological data, they have to put it in context, based on the temporal, evolutionary relationships between all of these species. The researchers at the Max Planck Institute sought to do their part in uncovering these secrets, and they looked deep into the cerebral cortex of reptiles and mammals, which shared a common ancestor some 320 million years ago. They first used data produced by one of the authors, Maria Antoinetta Tosches, in a previous experiment, which isolated transcription data from neural cells extracted from turtles and lizards. This transcription data is the mass of messenger RNA molecules that are expressed by a given type of neural cell. This messenger RNA reveals what genes are being expressed by what type of cell, and in what quantity, and this in turn determines the proteins that get expressed. It determines what that particular type of cell does, and where it resides in the brain. All of this transcription data was then used to turn an image of the cerebral cortex into a complex mosaic map, with each little patch in the mosaic being a clump of a particular type of neural cell. The researchers performed this modern study by comparing this intensely detailed map of the reptilian brain with equivalent data about the mammalian brain, and they found evidence of all kinds of changes and alterations that have occurred over evolutionary time. Specifically, they found differences in the hippocampus, which controls spatial awareness and the generation of memories. They found that in both reptiles and mammals, the hippocampus neural cells were near the center of the brain. But in reptiles, the structure was just a smooth, flat sheet that extended along the edge of the proximal sides of the cerebral hemispheres. Now in mammals, the hippocampus is all folded up. It's as if it's been squished, and it appears that, evolutionarily, it just may have been. Gilles Laurent was a lead author on the paper, and he said, quote, "It is as if, in early mammals, the ancestral hippocampus has been pushed by an increasingly dominant neocortex and forced to fold onto itself to acquire its signature mammalian architecture." Unquote. Where did this neocortex come from? Well, it exists in reptiles, but it's relatively small and undeveloped. It's still a thin sheet, much like the reptilian hippocampus is. But in mammals, this neocortex has greatly expanded in size, and in humans it's known to have 6 major layers. The researchers say, and I quote, "The transcriptomic signature of glutamatergic neurons in the reptilian cortex suggests that mammalian neocortical layers are made of new cell types generated by diversification of ancestral gene regulatory programs." Unquote. These glutamatergic neurons have receptors for a chemical called glutamate, which is an excitatory neurotransmitter in the brain. These neurons that receive glutamate are involved in excitatory pathways, which activate neurons involved in learning and memory formation. Basically, as mammalians diversified away from reptiles and lived in new habitats, like colder habitats at higher latitudes, They had access to new resources, but they were also vulnerable to new challenges and new threats that they had to adapt to. On an even more fundamental level, the mammalian metabolic rate accelerated, and they became endothermic. They produced excess body heat, and they could be much more physically active, but they also required much more food on a daily basis. As mammalian lineages responded to their increasing metabolic rate and their new habitats and new ecological niches, this led to, quote, profound changes of gene expression programs, unquote, in the excitatory neural pathways of the brain. And this makes sense, as these pathways would be excited as the mammals responded to all of their new stimuli and new stressors. As natural selection radically changed mammal physiology and life strategy and behavior over these evolutionary lengths of time, so too did it change the functional activity and the cellular structures of the mammal brain. Now, birds weren't a part of the study, but birds are another group of amniotes, and we have studies of bird brains that show that the regions involved in navigation are disproportionately large compared to their reptilian ancestors and to their mammal cousins. This makes sense for a species that has to undergo long migrations and that travels through the air, where they can see the landscape from this aerial perspective and they learn to navigate by recognizing landmarks. And this all goes to show how plastic and evolutionarily malleable the brain is. With regards to the study, the researchers also found that inhibitory pathways and GABAergic neurons that suppressed neural activity and regulated neural activity and neural growth, these showed no major diversification. There wasn't any new structures or new types of cells as there were with excitatory neurons between reptiles and mammals. This implies that reptiles and mammals actually share the same general architecture of inhibitory neural circuitry, which strongly suggests that these inhibitory pathways were present in the mammal-reptile common ancestor, which existed sometime close to 320 million years ago. And it's quite likely that this pattern of inhibitory neural pathways also existed in tetrapod ancestors that are even older than this. While this study is really impressive, there are naturally more questions that remain unanswered. Now that we see this similarity in inhibitory circuits, it brings up the question of whether GABAergic neurons, and for that matter, glutamatergic neurons, whether these perform the same function and have the same role in the brain in both mammals and reptiles. It might be the same chemical neurotransmitter, but it's possible that it has different effects on neural activity, and thus the animal's activity. This leads to the next challenge, which is finding the association between how the brain evolves and how the animal's behavior and life strategy changes, and what the relationship is between that physiological evolution and that behavioral evolution. Gilles Laurent was reported as saying, and I quote, "Our results tremendously clarify our understanding of the reptilian brain, and thus of brain evolution," unquote. Additionally, this method of using the transcription data to build a mosaic map of neural cell types is both a novel and a profound way to study the brain. Laurent says, and I quote, "There is a lot more to explore from these new molecular maps. This is only the beginning," unquote.